Hey Team Bio, welcome to your last screencast on the last of the big four. So this is the big four, part four, organic compounds. This is an organic compound. You might not have heard this name before. It's called nucleic acid. Oop. But I'm sure you've heard of DNA and RNA. Maybe not RNA, but definitely DNA. So there are two types of nucleic acids, DNA and RNA. And the functions of DNA and RNA, the primary function of DNA is to encode genetic information. So this is mostly DNA that's doing this, or it is DNA that's doing this. And this is kind of over the long term, or a DNA molecule is like a long-term storage molecule. It will exist for a long, long time, whereas RNA is a little bit more ephemeral. It's built and then broken down, built and then broken down. So RNA is short-term. Information storage. As well as some RNA molecules function like enzymes, so they catalyze reactions. And some RNA molecules modify gene expression. Modify gene expression. So, and this is all RNA. What this means is that RNA sometimes controls if a DNA gene is turned on or turned off. A big flow of information called the central dogma of biology is this idea that information is stored long-term in the DNA, and then it is transcribed into RNA, and then the RNA is translated into protein. So the DNA is the long-term storage molecule, and the RNA is the intermediary between the DNA and the protein, which is actually doing the business of living, catalyzing reactions and allowing things to happen in your body that wouldn't be possible otherwise. Okay, so DNA long-term, RNA more short-term, both involved in information storage and regulation. Okay, the monomer of DNA and RNA, they're both called nucleotides. So the monomer of DNA and RNA is called a nucleotide. So this is an example of one DNA nucleotide. It has a couple different important pieces to it. Number one, it's composed of a 5-carbon sugar. Number two, it has a phosphate group. And number three, it has a nitrogen base. These nitrogen bases come in 4 different flavors in DNA. They can be adenine or A, thymine or T, cytosine or C, guanine or G. So these 4 different flavors of DNA are the 4 different nucleotides. And I guess I'll do blue. Okay, so DNA has 4 different nucleotides, A, T, C, and G. RNA also has a sugar group. It also has a phosphate group. And it also has a nitrogen base. You might be wondering what makes DNA different from RNA. DNA stands for deoxyribonucleic acid, and DNA is missing a hydroxyl group on its sugar, on the 3' carbon, right here. You don't need to know that. RNA is the exact same structure, except it's ribonucleic acid. It's not missing that hydroxyl group. It has it. Okay, so they have slightly different structures. And then RNA has also 4 different nucleotides. A, adenine, U, uracil, C, cytosine, and G, guanine as well. So, what colors did I use? Okay, here. So red, as a highlighter. Adenine, uracil, cytosine, and guanine. Okay, so this is a single DNA monomer, but to create a polymer of DNA, we got to put a bunch of monomers together. So this is a DNA polymer. And when DNA forms a polymer, it always takes on this specific structure. So you'll notice if I took this nucleotide and I copied it over many times, so pasted it here, and then 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 I turned it 180 degrees and continued to paste it like this, this would give me a complete DNA strand. So DNA is composed of 2 strands. The backbones, the 2 sides, are held together in what's called a sugar phosphate backbone. Because if you look at what's on the outside of the molecule, you see phosphate, sugar, phosphate, sugar, phosphate, sugar. This is the same thing on the opposite side. This is also a sugar phosphate backbone. But you might notice that the 2 backbones are running in opposite directions. So DNA nucleotides have a directionality to them. This is called the 5' end of the nucleotide, and this is called the 3' end of the nucleotide. So if one strand of DNA is running from the 3' side is down here, this means the 5' side is up here. The other one is going to be running in the opposite direction, where the 3' side is up here and the 5' side is down here. So there are 2, as I said, sugar phosphate backbones. So here's one here, and then here's another one over here, but they're running anti-parallel to one another. So the fact that this is running from 3 to 5 in this direction, and this is running from 3 to 5 in this direction, we call this anti-parallel. Okay, so DNA strands are running anti-parallel to one another. Okay, the sugar phosphate backbone, I should also say, is held together by covalent bonds. So the bonds between the nucleotides, they're all covalent bonds that make up the sugar phosphate backbone. So when you look at the structure of the two sides of this strand of DNA, you see that the nitrogen bases face each other. So if this nitrogen base is, let's say it's adenine here, then this one is going to be thymine. And I guess I should say a little bit more about that. So DNA, in addition to running antiparallel, has a very specific pattern of binding. In a DNA molecule, if you look across from one side of the strand to the other, you'll see that A binds to T, and vice versa. So A always binds to T, T always binds to A, and C binds to G. The bonds that hold them together are hydrogen bonds. So the AT bonding pair has 2 hydrogen bonds, and the CG binding pair has 3 hydrogen bonds. Okay, so going back to our little shady bit, C is purple and G is blue. So if we were to go down the one strand of the molecule and just color it in, I want to do the highlighter with different colors. I know the sequence of this side is A, C, A, G, T, G. Then this side must be T, G, and then T again, and then C again, and then A, and then C. This is always true. A always binds to T and vice versa. C always binds to G and vice versa. So if you have one side of the strand of DNA, you can automatically infer the other side of the strand of DNA. And again, they're held together in the middle through hydrogen bonds. AT connection is 2 and the CG connection is 3. Okay, so DNA, we've said before, it encodes genetic information. And the way that it does that is through the sequence of letters, A, C, T, and G, and the order that they're in. You all know how to read, and you would know that if I spelled, let's say, dog, you would know what this word means. And if I rearrange the letters to go in another order, you know that now this has a new meaning. God is not the same meaning as dog. So the order in which the letters are in is important and conveys a different meaning. The same is true of DNA. The sequence of the nucleotides encodes information, and if you change the sequence, then you change the information that it encodes. And that is how our genetic information is stored. And more on this process later on in the year. Okay, really quickly, RNA. When RNA forms a long strand, a polymer, it always forms a single-stranded molecule. It still has a sugar phosphate backbone, so this is the same. And we would see the same, 3 of the same nucleotides in RNA, but one different one. So it will never have thymine. Instead, it will have uracil. So that's one major difference between, or I guess another major difference between DNA and RNA. RNA is always single-stranded, or I should say usually single-stranded, and it's going to have uracil in the place of thymine, but it still has cytosine, adenine, oops, that's cytosine, and guanine. Okay, that's all. See you in class tomorrow.